In most science programs, students are required to carry out a final year project where they are exposed to the steps in planning research, designing their projects, experiments, collecting data, analyzing them, and then presenting the data, and also to write out a thesis. So in microbiology, which is the science of microorganisms, of course we study microbes, whereas the definition of science is the observation, identification and description of natural phenomena. So after that, experimental investigation is usually required in order to try to explain the phenomenon that was observed. And to do the experimental investigation, you have to be familiar with appropriate research designs, how a researcher can systematically design a study to ensure valid and reliable results that address the research aims and objectives. So when you look at research designs, we can divide them into experimental design and also correlational research design. So in experimental design, it is more of a fixed laboratory or ex situ condition where you manipulate the conditions to test out independent variables. So it is a laboratory condition where the disadvantage is the results will never be able to replicate real world situations. Next, for correlational research design, it is usually of exploratory. It is more exploratory in nature. Usually we a monitoring exercise is part of the exploratory research. Results are obtainable and usually a wide array of variables are measured in order for us to be able to come to a conclusion. Okay. Usually analysis is carried out via correlation to look for patterns, to look for trends. However, correlation does not equate causality. So just because you observe variables that correlate to each other does not necessarily mean that one is causing the other. Although correlation is necessary for causality, it doesn't equate causality. For me, the best practice is always to do both. Okay, either a correlational research, exploratory in nature first, to establish a hypothesis, and then to carry out experiments where you can manipulate the variables in order to test your hypothesis. That will be a very good research design. Other labs carry out laboratory experiments first, where they look at certain parameters in order to come up with suitable hypothesis, and then they look at nature to see whether their results are applicable in the natural environment. Okay. So what is the role of statistics then after you have carried out your research? So statistics is the discipline that concerns the collection or sampling, the organization, analysis, interpretation, and presentation of data. So for presentation of data, it's usually graphical output, how you present it. All this requires exploration. You have to try out the different types of graphs and the suitable graphs that are available. So here I would like to stress the difference between statistics, which is any value generated from a sample. So what is a sample? A sample is a subset of population. So it is impossible for us to study the population at large. Almost always we only sample a subpopulation, a subset of the population. So sampling is important because you have to ensure that your sample represents the population. Okay, and then any value that you generated from the sample is called statistics. This is in contrast to a parameter, which is any value generated or applied to a population. So such as population mean or population variance. Next, we will go more in detail into statistics and the actual practice of carrying out statistical tests.